Bismillahirrahmanirrahim. My name is Momina Amin, and the topic of my presentation is bioinformatics computing. What actually bioinformatics computing is? In bioinformatics, we solve biological problems and process biological data using computational techniques of mathematics and statistics. We use computers for different purposes in this field. For example, number one, storage. For example, we get some kind of information from any experiment we have done. So we have to store that information. That can be any genetic sequence, DNA sequence, or any biological information. So we can store it in our computer. So actually, all these informations, all these kind of sequences are so huge that they cannot be memorized or handwritten. So we store them in our computers. Number two, retrieval. For example, we need that information back. So we can access that through our computers, so we can retrieve it. Number three, manipulation. For example, we need to edit that information or make a change in that information. We can do that whenever we want. Number four, distribution. Now, we can distribute this information into any kind of classes whenever we want, using our computers for our convenience, using the formulas already present in the computer. And all this information in the bioinformatics we deal with is related to DNA, RNA, and proteins. Bioinformatics computing is also called as computational microbiology. In bioinformatics, we have biological problems we deal with that we solve using mathematics and statistics, their operations and logics, with the help of computer science. In all these forms, bioinformatics, so biology, maths, computer science, all together form bioinformatics. Now, what actually bioinformatics data is? In bioinformatics data, we deal with data related to number one, DNA and protein sequence, as shown in the figures below. Number two, gene expression. Now, what actually gene expression is? Gene expression is a process in which we use gene information for the synthesis of functional gene product that ends up as a protein. Number three, raw data collected from any fields or laboratories in which we have performed the experiments. Number four, this data can be related to images, can be images or virtual models, images of the things that are known, their functions, their sequences, they are known, and they can also be virtual models of any product that is unknown but is predicted by the scientists. Number five, they can be articles, books, or publications, and they can also be any theory that is predicted. Now the components of bioinformatics are, there are three main components: creation of database, development of algorithms and statistics. Number three, analysis of data interpretation. Now in creation of database, we have to organize the data first we have gained through the experiments we have done. And then we store it in our computers, and then we manage it into biological data sets. In the development of algorithms, we first of all make algorithms and tools that can be used to determine the relationship between the large data sets members. So we can compare the protein sequences, sequence data that we have and that already existed of the known protein sequences. So through that we can find the relation between them. And the third one is analysis of data. So we analyze the data and after forming the relation, we conclude or end up with some kind of information that we need. So for example, we end up with protein sequence or a DNA sequence or structure. Now, what is the importance of bioinformatics? It has huge importance in our lives because it lets us know the important pathways and networks of the biological systems that make us alive. Now, it helps in a human genome project by identification of disease-causing genes. By that, when we come to know about the disease-causing genes, we can also treat them by creating the remedies. Now, they can also predict the those patients at the risk of high risk of that disease. Now, if this can also help in the field of medicine, as they can produce drugs for that medicines, the personal medicine for some specific person or a specific purpose, predictive medicine to predict and gene therapy for that can be also called as gene expression. It improves the diagnosis and is also prognosis of many diseases. Now, impact on our society, it has a very large impact on our society. First of all, we're going to talk about pharmaceutical industry. In pharmaceutical industry, it can be used to identify and analyze drugs, and also can be used to produce new drugs in the field of biology. And it can also be used to increase the potential of drugs. Number two, in agriculture. It has a very large use in agricultural field. We can study the genes of agricultural species, and those genes can be those species can be modified so that we can for our benefits. Those species can be animals, plants. By modifying them, we can improve the production of agricultural products we get, for example, meat and milk, and produce high yield crops. And they can also they can also need low maintenance. Number three, drug discovery. So it can also be used in the field of drug discovery by producing new kind of drugs, by drug development, and developing those drugs in a small amount of time, in making drugs cost effective by using such products in them, or that are not very costly, or not using very costly ingredients, or making them ourselves. It can also help in environment because it is used to identify waste and clean up bacteria, and can overcome our pollution problems. And now, fields related to bioinformatics, there are lots of fields related to bioinformatics. Drug development, we can develop drugs. Gene therapy, we can use genetic information for the synthesis of some functional gene that ends up as a protein. Microbial genome application. Crop development, we can develop crops of high yield with low maintenance. Molecular medicine, forensic analysis of microbes. Biotechnology and evolutionary studies. In evolutionary studies, we study the genes before the evolution and after the evolution, and we compare them to know what actually caused the evolution. And last but not least, bioinformatics. Now we can go through the brief history of bioinformatics. It was for the first time introduced in 1990. However, it was all and there in the past, but for the first time as a subject, it was introduced in 1990. In 1859, Charles Darwin gave the theory of genetic evolution. In 1869, DNA from nuclei of white blood cells were isolated for the first time. In 1951, alpha helix and beta sheet were discovered. In 1953, Watson and Crick double helix model of DNA was discovered through X-ray. In 1955, first protein sequence was announced. In 1958, and so on till now, different organizations and projects have been launched, like ARPA, PDP, PERL, NCBI, Human Genome Project, DIGR, Human Genome, etc., to enhance this field. Now, there are so much applications. We can apply the bioinformatics in so much places. So number one, we can use this to find the sequence mapping of biomolecules, for example, DNA and RNA in proteins. And we can also use it to identify the nucleotide of a gene and the sequence of functional gene. Number three, we can use it in finding the sites that can be cut by restriction enzymes. For example, when we have to optimize the working of something, we attach enzymes with it. So we can find those. These enzymes are attached to some specific sites, so we can find those sites by using bioinformatics. Prediction of functional gene product. So, what is functional prediction of functional gene product? In this prediction, we have we do this mostly um, by gene expression, in which we have first we only have a sequence we have found through some experiment, but then we, using computational techniques, find the structure of that sequence, and then we compare that structure of unknown gene with a known gene that is somehow related to it, to predict the function of that gene. The market worth of bioinformatics. In 2019, the market worth of bioinformatics was 7.32 billion US dollars, and it is expected to be 20.55 US billion dollars in 2026. Freelancing, it has a huge scope in freelancing. So actually, we in freelancing, one is self-employed and boss himself. So in freelancing, one can produce tools and algorithms to help further in human healthcare research, human gene editing, genomics, metabolism, etc., etc. And number two, we can execute pipelines of RNA sequencing, genome, exome, and next generation sequencing to determine expressed genes in some conditions, co-expression networks, distinguishing between subtypes, predict drug sensitivity of cell lines, and so on. There are so much things to be done in the freelancing, but these two are the main ones. And now the most important thing is how to enter this field and which subjects should one choose to enter bioinformatics. So majority of a bioinformatics scientists told that they entered this field after bachelor's degree in mostly in the subject relating to biology. For example, biology, biotechnology, biology sciences, molecular biology, photobiology, radiation biology, and biochemistry. Now, what is the level of degree recommended to enter this level? So it should be bachelor level. It is actually bachelor's degree level, because you can do master's, PhD, and postdoc, and so on in different courses related to this field after getting into this field. Thank you.